Good morning, everybody. Welcome to day 4. It is day 4, isn't it? Yes, oh good. So we've talked about all kinds of subjects on route to today, but I feel like in a lot of respects, the subjects you learned yesterday about DNA sequencing, mapping, and assembly, and the things you learned today about gene expression are kind of the core body of technologies everybody is expecting you to know in the space of bioinformatics. There are a lot of other things that fit under that helm, but that's a very, these 2 days I think are some of the most essential bits of bioinformatics that everybody should really understand. Today we're going to move away from just thinking about sequencing per se, and instead start speaking about gene expression. How do we evaluate which genes are active for a particular tissue or cell or whatever? We're going to start with a bit of a primer on why measuring this, the transcription process is valuable. And for that, we're going to talk about the technologies that we use for it. I think a lot of people imagine that microarrays are passé and that their day is over and that no one uses them anymore. But I would point out that a huge number of them still sell every year. And there are a lot of good reasons why microarrays continue to be one of the easiest ways to deal with gene expression data. From there, we'll move on to talk a little bit about RNA-Seq, part of the massively parallel sequencing entry into the space of transcriptomics. And then from there, we're going to move into quite a bit of talk about data analysis. We're going to talk about biclustering, which is one of the key technologies that we use for visualizing microarray data, or RNA-Seq data, both get shown that way. And we'll certainly talk about difference testing. We'll discuss something called the volcano plot and the MA plot and different things like that. Because when you read a paper about gene expression, you're sure to see one of these plots, and I want you to be able to interpret it when you see it. And of course, I am enough of a statistician, I'm a pretender in this space, but I feel strongly enough about these statistical issues that we're really going to have to talk about multiple testing correction, which is a key topic that a lot of people fail to do right. All right, so why is gene expression such an important target of regulation? We see that gene expression is a regulated process because mutant cells versus wild-type cells may exhibit very different behaviors due to even a single mutation. We see that which genes are being transcribed can change in response to stimuli, such as drugs, light, or sleep. Did you realize that you could express different genes depending on whether you let yourself sleep 4 hours last night or 8? It's true. At different developmental stages, we have different genes growing. A fetus is going to use sets of genes that may be relatively unused much later on in life. So that's quite important. Different cell types. Obviously, if I have my muscle cells versus my skin cells, they have very different jobs. They don't need to express the same genes. And certainly in a state of disease versus health, you're going to see very different regulation. This is very frequently seen in diseases like cancer, where the amount of dysregulation is extreme. So generally speaking, we're hoping to measure the number of messenger RNA copies in a cell for a gene. And we think of that as an indicator of how much of the gene product, in this case the protein, is present. There are a lot of weaknesses to that assumption, though. So I would stress that when I talk about transcription, I'm talking about how much messenger RNA is being elicited from which set of genes, not so much how much protein I think is there, because measuring protein separately is really something you must do to assess those levels. I think the next thing that I think is worth talking about is that people often assume that if you sequence something, you know everything you need to know in order to evaluate its chance of disease or its phenotype or whatever. This is clearly wrong. It's clearly wrong. The genome, although it's the same genome throughout that organism, most typically, does not tell you all that much about what's happening now for that organism. It tells you what it's capable of. What's the way I always describe it is it's the catalog. It's a catalog from which these cells can order products. But just because something appears in the catalog does not mean it's currently being ordered. Do you see the distinction there? Transcriptomics more or less reflects the set of invoices against that catalog to say, well, the cell is investing in the purchase of this product and this product and this product. But they're not actually actuated until the protein itself is produced from it, in the case of protein-coding genes. Now, you can say if this protein is present, I think this process is taking place. And yet, some proteins may be present and yet not in active form yet, right? It might be that the protein only goes into action when it's phosphorylated, when, for example, epidermal growth factor has arrived and stimulated this particular receptor, which kicks off this phosphorylation cascade. So even the presence of the protein may not be enough to justify that this activity is present in the cell. Metabolomes themselves, the metabolites themselves, may be your best indicator to say, well, the presence of ornithine is diminishing as the presence of this other product made from it is rising. So we think of this as kind of getting closer and closer to what we used to call the coal face, to say that the dude working in the back office has less to do with what's happening in the mine than the miner does, right? Genomics is several steps removed from what's happening at the metabolomics stage. Now, I think everyone remembers that how the central dogma basically works, but I want to remind you of some really key differences between prokaryotic and eukaryotic gene transcription. So let's start with the fact that when we look at sets of related genes that are part of a common pathway, very typically, we see that the bacterium frequently has those organized together in an operon. So multiple protein coding genes that are all lining a pathway are all expressed conjointly as one big transcript. And we see that there are different ribosome binding sites on this transcript where different products may be produced. If a ribosome binds over here, then this first product is translated. If the ribosome binds over here, then the second product is translated. So that's quite different than what we see in the context of eukaryotes. Eukaryotes, on the other hand, have this intron-exon structure to their transcripts, which means that one gene may result in a variety of transcripts. In this case, we could imagine that exon 1 and exon 2 and exon 3 are all included in the transcript. That's what's visualized here. But there may be circumstances under which this cell skips exon 2 to say that exon 1 and exon 3 become the transcript, thus a different product than what would occur in the case where all three are used. We do not typically see scenarios where one transcript results in multiple multiple protein products in this case. But the intron and exon structure, it leads to a great change in how many potential transcription products there are versus the number of genes, the protein coding genes in the genome. So in general, just to fill in some numbers on that, we generally think of the number of protein coding genes for humans as being around 20,000. I think that my number might be a little high now, the number dances up and down from time to time. So about 20,000 protein coding genes in a human. But the number of transcripts that we can produce, I think, is probably in excess of 100,000. Right? That's 5 to 1 already. There are a great many genes that can be spliced a great many number of ways. Okay, so that term I used, splicing, is to refer to which pattern of exons are conjoined together to create the final messenger RNA. Okay, now RNA is twitchy. I don't know if you guys have heard that term in connection with messenger RNA, but it's twitchy. If you look at it wrong, it might degrade. So we don't like working with messenger RNA directly insofar as we can maybe make it more stable by producing complementary DNA from it. So complementary DNAs means that we want to create a DNA complement to a messenger RNA strand. Is that something that our cells inherently do? It is not. So in this case, we are borrowing a viral protein for making an RNA-dependent DNA polymerase. Everyone hears the name there. So we are reading from an RNA template and building a complementary DNA sequence from it. So that's called a cDNA. And cDNAs are very, very much nicer to work with. We don't have to worry about a stray RNase coming by and degrading a cDNA. That's not going to happen. Now, it is possible that you could get a DNase in there, and then you'd be in a real bit of trouble because the thing you just created would get chopped up for you. That's not going to be very helpful. So you can imagine that you may have you're almost always going to find that our messenger RNA measurements are made from a cDNA instead. And this gives us an opportunity that we can amplify the cDNA that we have in our sample before we go about measuring it. I don't know if we have audiophiles in the room, but occasionally when you go to some public event with people who don't know very much about sound systems, you'll find that they pre-amplify the signal that's going to the speakers. And as a result, you end up with this blah, blah, blah sound coming out of the speakers that's not very easy to understand at all. This amplification process is something that can happen here as well. If you rely too much on amplification prior to quantitation via RNA-Seq or by microarrays, you may distort the original proportions of the messenger RNA collection. So we need to be careful about how much amplification is used in advance of the subsequent measurement of the cDNAs. All right, so from here we're going to start talking about the technologies that we can use, in this case microarrays and beads and our RNA-Seq. So we speak of the, I know if you go into PubMed and do searches for how many different papers use the word proteome, you get a very large number. If you enter the word genome, you get a very, very large number. But it seems that a lot of people have forgotten about the existence of the transcriptome. The gene expression very frequently has kind of been left behind in the terminology of our field. You will see it called functional genomics. You will see it called gene expression. But the transcriptome per se is something that we speak about a little less often. So I think it's worth introducing the term here and reminding ourselves that it exists. Many of the techniques are based on hybridization. Does everyone remember what hybridization is? Oh, that's good. Okay, so we have two different strands, and if they share complementary sequences, hydrogen bonds can form between these complementary sequences to hybridize one sequence to the other. That's an important concept. So if you're using a microarray for this, then you have probe sequences on the surface of the microarray, and if they are complementary to a cDNA built from your messenger RNA, then they will hybridize there. If these arriving molecules of cDNA are labeled fluorescently, then where hybridization occurs, you will have some fluorescence that you can read as well. That's quite different than a sequencing-based approach. In a sequencing-based approach, you are actively sequencing the cDNA molecules themselves made from the messenger RNA. That is very powerful, and yet it's a completely different sort of readout, isn't it? Because we think about light intensity as being continuous, right? We have some amount of background, but if you have enough fluorescent molecules together, you get a signal that's perceivable above that background, and it rises and rises and rises as you have more and more copies of cDNAs there. Sequencing is quite different because now we are counting how many of our reads corresponded to this particular gene product. Right. So that is a sequencing-based approach. That's the RNA-Seq route. Now, the way that we make DNA microarrays has changed a fair bit over time, but we're going to start by just defining what the DNA microarray is. Does everyone remember Cartesian geometry? Cartesian? Okay. Cartesian is named after René Descartes, who was a mathematician and philosopher. A Cartesian array, in this case, is an XY grid. So if you have an XY grid, you have these little spots in a grid. Each of those spots is a different Cartesian coordinate. At these different spots, we're going to have a whole cluster of probe sequences, and these probe sequences should all be identical for that one spot. If you have the ability to make these spots very, very small, then you can pack a whole lot of different features, a great many, millions in fact, of different probe sequences on a single XY plane. That's quite powerful. So whenever someone says Cartesian array, I just want you to think of an XY plane with something appearing at each grid point. Now, we have kind of an early approach that was used, and one that I think is somewhat less common now, which is the spotted or printed array. Does everyone remember inkjet printers? Few people? All right, well, an inkjet printer is one that sprays ink onto the page in order to create the letters that you print on, right? So they were ridiculously cheap, ridiculously commonplace in the States at least. I think they sell quite a few of them here too. So these inkjet printers have a very precise ability to deposit a certain amount of ink, often of multiple colors, on the page at a particular spot. You can use that same kind of technology to instead of spraying ink on a paper, spray DNA onto a particular spot on a membrane of some sort. Right? So you can create these collections of probe sequences at these very closely spaced locations on this membrane. That is the early way to make a spotted or printed array. These days, you see a lot more people making use of synthesized arrays. And this is a case where we build our probe sequences in situ. In situ. Do people know what in situ means?
Yes.
In place. In place. Oh, it's over here. Now the technology that we use for that is nothing short of miraculous, if you ask me. Does everyone know how we build microprocessors? Well, we're all going to learn something fun then. Photolithography. Photolithography. I have three parts to this word. Photo, which means what? Light, good. Litho.
Chemical or something?
Something like stone, okay? Photo, stone, and graphy. Graphy. Writing. So with light, writing in stone is what that means, photolithography. We have the ability to build transistors on a silicon surface. And in the same way, we can build DNA on these surfaces. So this is a really amazing thing. Imagine that we have this surface on which we're going to write a microarray, and we cover it with a mask. Wherever this mask is, stuff is not going to happen. Let us then say that we know that we want to add adenosine to this spot, this spot, this spot, and that spot, but not other spots. A laser now comes along and burns a hole through the mask at those locations only. We then add A and some polymerase, and it synthesizes an A wherever the mask has been burned away. Now we remask. Okay, so once again, no reaction can take place unless we burn away that place. Next, our computer has decided, I am going to add C wherever C is the right place, is the next residue to be added. Those locations. And then we add C, it's polymerized to that the growing probe sequence on the surface. We remask, and this time maybe we say G is what we're going to add next. And then we add G to just those locations. It sounds too weird to be true, but in fact, photolithography is the way that many people manufacture probe sequences right on the surface. You can imagine, for example, a probe sequence of 25 nucleotides is built by 25 different layers of unmasking and polymerization on that spot. How could this ever work? And yet it does. This ability to do photolithography allows us to create very, very dense microarrays with spots that are just a few microns across. It's an astonishing technology. All right, now, as things have moved along, people have kind of moved away from the idea that I want to... has anyone ever heard of a U133? Okay, well, for a while, this was like the go-to array in the space of human gene expression. If you were going to check gene expression, in this case, gene variance, this was for sequence variation rather than expression, I think. But they would use this in all kinds of these assays to power GWAS studies. We're not really going to talk about GWAS this week, but if you take the NGS unit, we will. So these days, instead of expecting that everyone uses the same kind of microarray because everyone's doing the same kind of research, you frequently see rather custom types of arrays used. Now you remember a couple days ago we were talking about the degree of genetic diversity found within Africa, right? If you were to use a genetic diversity microarray that was really tuned for European or American populations, you would be missing a whole bunch of genetic variation that's available for evaluation in an African population. So if you were to use one of those microarrays, you would be missing out on half the story for African populations. So these days, we see a lot more people customizing arrays. So the H3 Africa effort that's headquartered over at Cape Town has designed a type of bead array rather than microarray, an array built from beads that will allow us to sample far greater genetic diversity that's present in African populations that may be completely absent in a place like where I'm from. Okay, so let us imagine that in this case we do not have our Cartesian grid anymore. We don't have the probes built on this particular spot on a plate. Instead, we have a bead that's covered with a lawn of the same probe sequence. All right, so when I interrogate this bead, I can get two kinds of information out. First off, which sequence does it represent? And secondly, how much does it have in this lawn covering its surface? But to measure these things most conveniently, I could use something like microfluidics, but more typically, in this case, we use these wells that are spaced, in this case, at 3 micron, to house all these little 3 micron beads that are covered with the lawns of probe sequences. So if you can force the beads to sit in an orderly way like this, you can then measure each one and say, okay, which probe sequence do you represent? How many copies do you have attached to you? Okay, next, you, which probe sequence do you represent? How many cDNAs are bound to you? Kind of shocking and amazing. So bead arrays have come to be not necessarily a replacement for microarrays, but have become a much more flexible collection, so that if you have a particular set of beads that you want to include in your array, the companies that manufacture these beads can put together these different collections for you to make a bead collection that's appropriate to your research. A little bit more flexible than simply manufacturing a one-size-fits-all genetic array, for example. Okay. Now, a lot of people have shifted away from the use of microarray data, and they've moved to RNA-Seq instead. RNA-Seq, as I mentioned, does not work on this hybridization principle to say, I have this canonical probe sequence, how much is bound to it? Instead, they're working on the sequencing directly from the cDNA molecules that have been generated from a messenger RNA collection. All right. So I wanted to try to talk about 3 different approaches that have been used for making sense of RNA data. So let's start with the one that has the most information coming to it. In this case, we have some genome reference, and from that genome reference, we have gene models that have been put forward for this particular species. So this gene is known to associate with these 4 different transcripts that can all be built from this genetic sequence. Everyone sees that? We already talked about intron-exon splicing. Here we have 4 different models of how that splicing takes place for one protein-coding gene. Now, from that, we have the interest of knowing which of the known transcripts for this species have been observed in this RNA-Seq experiment. And these little blue bits represent reads. So some of them fit all the way inside one of those exons. This one is a full exon, full exon, full exon, etc. These are all complete exons that are found within this last part. But you see these that have this line run through the middle there? That is to reflect that this read starts in this exon and continues into that exon. So that's really powerful. In this case, we may be less interested in knowing which reads map entirely within one exon and more interested in knowing about those exons that bridge between 2 exons, because that's rather informative about which isoform is there. If we have a read like this, we know it could not come from one of these first 3 gene models. Do you see that? Because these all have this exon inside. Stepping out of this exon would step into this one. But because these step from this exon to that, we have great assurance that this is it has to be an isoform, a transcript from this gene that represents this exon and this one, but not the one in the middle. Powerful. Now imagine that you have somewhat less information than that. Maybe you feel that the set of transcripts known for this species is rather insufficient. That you have some notion of which transcripts are there, but you feel pretty sure it's incomplete. You might use software like Star Aligner to say, I have a genomic reference and I have these reads that I've produced. Am I able to put gapped alignments where one read can be mapped to make perhaps very distant parts of the genome that give me evidence that the gene expression spans from this space to this space? How far apart are features for a given gene in the genome? If I have a protein coding gene, is it all going to fit within a kilobase? No. Not within a kilobase. That's way too small. If you're dealing with transcripts that are a kilobase that are all together, you're probably dealing with a microbe, right? But if you have a human genome, it's quite likely that the exons are relatively small compared to the introns that separate them. You can have as much as a megabase separating this exon from its next one. So being able to do this alignment across these very large gaps that may occur due to introns gives us the ability from RNA-Seq to greater document which exons come together to form these unions. So in this case, we have both an annotation, the original sequence, and the annotated isoforms from a gene. In the next, we have the reference genome, but not the gene models that would be applied to it. Sometimes you end up with something really crazy, a total non-model organism. And in a case like that, you may not have a genome to compare against at all. That's the case shown over on letter A. In a case like this, you may be trying to assemble transcript sequences without even having a genome to compare them against. This is the scenario we find ourselves in with our hyena project. So in a case like that, you don't have the genome reference to work from, you're just stitching together these transcript sequences as well as you can, given only the cDNA sequences derived from messenger RNA. Okay, so that's three very different kinds of applications, from the case where we have a lot of information about this organism to a case where I have almost no information about this organism. I've included this slide in here because it's a useful reminder that these workflows that I'm talking about have a lot of different steps to them. I have mentioned Burroughs-Wheeler alignment algorithms. I think we talked about those yesterday under mapping. So BWA is the basis under which Bowtie operates. So you can see that if you have a list of known transcripts from the species, you can perform ungapped alignments to these transcripts in order to determine how much each transcript has been observed. It may be, however, that you really have zero information to work from. In a scenario like this, you might want to try software like the Trinity de novo assembler. Trinity is a pretty amazing piece of software. I've actually had some experience working with this in non-model organisms. And given a pile of RNA-Seq and no genome annotation, you can get pretty close to a lot of transcript sequences that are quite real. So that's quite powerful. The blue set at the left here represents the case that was at the most extreme right in the previous slide. So I want you to keep in mind that when you see this slide and this slide, the workflows are presented in the opposite order of what they are in the slide before. So when you have the greatest amount of information and you're doing a gapped mapping against known gene models, something like the model that we see at the left is best. Okay. This brings us to data analysis, and I'm going to suggest that we take a very quick break and then come back in here. Welcome back to the second part. We're going to talk about the data analysis of RNA-Seq and microarray data experiments. So we have this problem that what we are reading is fluorescent intensity in microarrays, but we don't care about fluorescent intensity per se. What we care about is how many molecules do we have at that spot. So in general, we're working under the assumption that if you have more of the sample cDNA hybridizing to the probe sequences, you're going to have a brighter signal to work from. Dim spots have more error. This is not unique in the case of microarrays, but in most cases when you have vanishingly small amounts of some transcript that you're trying to measure, you're going to have a larger degree of error associated with it. In general, we see that software like RMA, the robust multi-array analysis, gives us the ability to deal with variations from array to array. Right? So it's quite typical these days that you would use one microarray for each sample. If you've got 50 people in your case and 50 people in your control set, that's 100 microarrays that you need to normalize across, that you need to be able to compare an intensity seen in this array to an intensity for that same transcript in another array. So something like robust multi-array analysis is a pretty good way to go for doing this kind of thing. When they compared to some of the approaches that were initially put forward in this space, they found that there were some really serious problems. As one gets to lower and lower expression, that we had greater and greater standard deviations of variance, of standard deviations among different replicates. Now, how do we translate a number of reads that we've seen for a particular gene or a particular transcript in RNA-Seq into a number that we can reliably use? Imagine that you have a whole lot of this sample and a relative paucity of this sample, and you've run both of them through RNA-Seq. The numbers will be higher in the sample where you had a lot more sample to work from than in the one where you had a vanishingly small amount of sample. So being able to normalize would probably require that you have some way to adjust for the fact that you had more sequencing reads in the first case than in the second. So when we compute how much we have of a given set of exons, we typically see something like RPKM used. So I want to try to walk through this expression that we see at the bottom. The number of reads is simply the number of sequencing reads that were mapped to this particular transcript. Okay, so we're not asking about total number of reads overall, we're asking just about the reads on this transcript. Gene length is the length of this transcript sequence. So imagine that you are looking at a typical transcript for humans. That's something like 2,000 nucleotides long. So a transcript that's 2,000 nucleotides long is more likely to have reads mapping to it than one that's 1,000 nucleotides long. A transcript that's 4,000 or 5,000 nucleotides long is going to have more opportunity to produce reads than one that's much shorter, like 2,000 or 1,000. So we must be able to take into account how big a target we could potentially have pulled reads from when we try to say how much of this transcript is there. And finally, the total number of reads matters. So if you had a very sensitive sequencing experiment that you're combining with data with samples where you had a relatively insensitive sequencing run, you need to be able to take that into account. The total number of reads takes that in. So the RPKM stands for reads per kilobase of exon model per million reads, which means that we need to divide the total number of reads by 1,000,000, and because we're talking about kilobase of gene model, we need to divide the transcript length in this case by 1,000. So when the values that come out of RPKM are going to give us a way to adjust for the fact that the sizes of transcripts can vary and that the sensitivity of sequencing in general may vary. All right, now I've already said normalization is a valuable thing, but I think I may have kind of hoped everyone would just get what that meant. I don't think that's completely fair. So let's talk about normalization again. Imagine that we are looking at 8 microarrays, each represented by a different box plot here. I think most people have encountered box plots, but if you haven't, I'll just say that the bottom of the box we typically use to estimate where the 25th percentile of the data are. And then we have the top of the box that's representing the 75th percentile of values, and then the middle of the box is the median. Then we've got some spray of data that fall outside that within the whiskers, and even some that fall outside that. So when we visualize the data in this way, we are not going one gene at a time. Each of the genes is somewhere within that box plot. So we expect that, in general, the bulk distribution of intensity values that we see of strength of transcription at these spots is going to fall in roughly the same distribution from array to array. But we see that that's not strictly speaking true. Do you see how box number 7 on the without normalization plot has a higher top edge? That's to say that its 75th percentile of intensities is a lot higher than all the 75th percentiles of the other 7 experiments. So is that a problem? It could be a problem if you always see a higher expression value for all genes characteristically for sample 7. If the statistical model you're using presumes that they're all distributed roughly the same way. So one of the most straightforward ways that you see people deal with this is to find a way to make the medians match. You see that the middle lines on all these boxes are at the same horizontal level here. Same vertical level here. So in this case, they are using a constant that they multiply each of these by that forces the mean value to 1 in this case. So they compute the average for that microarray, and then they divide by that average. Everything now falls to, so that the average falls on the vertical value 1. You can see that right here. So that's a global normalization. It's kind of a blunt instrument, I must say, but it seems to work reasonably well. There are times, however, when the statistical models you're working from really need the data to be distributed the same way, quite uniformly across all of the microarrays you're combining. And for something like that, a quantile normalization approach might be appropriate. This is a case that helps us to avoid distributional changes from microarray to microarray creeping into our statistics and making us make mistakes. Okay, so normalization is a way to ensure that all these different experiments have the same representative level across them. Okay, now there's a lot of QC that frequently takes place in microarray experiments, and that is to protect us from bad things happening. So the Bland-Altman approach, or the MA plot, is one of the most common kinds of representations that we use to ask whether a given pair of microarrays can be compared or not. So I want you to think of each gene as represented on 2 different microarrays being shown somewhere on this cloud of dots, all right? So each gene is now represented here, and we're doing a comparison, one microarray plate against another microarray plate, or one RNA-Seq experiment versus another RNA-Seq experiment. In this case, the dots are going to fall on the 0 line vertically if there's no fold change difference, basically. If you have a 1-to-1 fold change for this gene. The dot is going to appear to the left if it's of low intensity, and it's going to appear to the right if it's a high intensity. So these are genes for which we had a screaming loud amount of signal. This is the abundance here is shown on a log scale. So these are just incredibly abundant. These down at the bottom are just very, very weak signals. Now, some of the stuff is going to be way up, and some of the stuff is going to be way down. That's okay. In the main, we expect the great bulk of genes to show roughly the same abundance between these 2 scenarios. So one of the things that we would ask is, do we have a skew in the fold change for a given gene that's a function of abundance? So that might seem a little weird. Let me try to explain that. We already mentioned that we're more likely to have high variance in our measurements at low abundance levels. Do you remember our talking about that? Basically, if you have very few copies of a transcript, your ability to quantify it is less by a large factor. Having lots and lots of that transcript makes it a whole lot easier to detect significant differences. All right, so let's imagine that in this plot, we had a characteristic big skew where almost all of the values had positive M, had positive fold change ratios, log fold change ratios, I should say. This is not I was talking about this line as a 1-to-1 fold change. In other words, no change in concentration. It's at 0 because this is a log fold change plot. A log fold change. So a positive value means more in A than in B. and a negative value represents more in B than in A. Okay, so you can imagine then that this plot gives you at a glance a pretty good appreciation of whether the data are centering on the no change line as a function of abundance. This is what things look like when everything goes out the window. So in the case of the left, we see that at very low log abundance levels of this comparison between plates, we have a very low typical log fold change number. We drop down to -1. If that's on if that's log base 2, it means that the typical performance at this low abundance is that we have a twofold change in one direction. That's very frustrating. People would not really like to include data of this sort. It is possible then to create linear models that will readjust these things so that the parity, the log fold change, stays at 0 as a function of abundance. So this process of normalization can fix these errors for us. You would prefer that you don't have to fix your data, right? If you have some samples that are so clearly distorted compared to others, you should probably rethink rerunning those, just saying. Okay. So, batch effects. Batch effects is something that you'll note I've put capital letters on batch and on effects. Batch effects, big deal, great concept for people starting their research careers to have this term well defined in your mind. Instrument performance varies with time. This is not a controversial statement, but it can fly in the face of people who are accustomed to thinking that if I spend $1 million on an instrument, it's going to give me great performance all the time. This is not true. Instrumentation has all kinds of weird twitchiness about it. You might find that your mass spectrometer gives you different results after they paint the hallway outside your lab than before. People say, oh, well, if I just sequence the same sample repeatedly, I'm always going to get the same result. You're not. Sequencers, microarrays, mass spectrometers, confocal microscopes, whatever you name it, all of them have good days and bad days. And batch effects are a result from that. So I would like you to just, the easiest way to take this in mind is to think to yourself, what if I do all of the cases for my study this week? And then I'm away for the next couple weeks, and then I come back in at the very end of March and run on the instrument, and this time I'm going to do all of my controls. This is a terrible, terrible thing to do, because it will mean that you have a conflation, that you are involving in your study that differences between the case and control cohort is not just the biological difference you wanted to measure. There's also a difference in whether the instrument was on the good week or the bad week, and you don't know in advance when that was. So, being able to manage batch effects is something that becomes very necessary in studies like that. But generally, those are not rescuable, right? You can't just fix that. You can't hand your data to a bioinformatician and say, get rid of the batch effect for me. It doesn't work like that. We don't have the information to do so. So, if you must acquire your batches, your samples in different batches on the instrument, there are some ways you can deal with that. First off, clearly you should randomly distribute samples to batches. It may simply be that you'll only be able to do 50 this week, and you must do in total 150. If something like that is necessary, at the very least, randomly distribute the samples from cohorts to when they're going to be run on the instruments. Inform the statistical model you're using that these experiments were produced in batch 1, these were produced in batch 2. This is a covariate that may help your model disentangle this batch effect. And it's always helpful if you have the same sample run in every batch, because that will give you some sense of just how much variance is occurring because the sample was being run at multiple times. I want to say that everyone knows about batch effects and everyone is always very careful about avoiding them. But I have to say there are some very well-funded, very prestigious groups who do some really bad science and don't know it. Keith Baggerly is sometimes called a forensic biostatistician. He likes to look into these studies and discover where things went wrong. When he's found data that look a little suspicious, he tends to jump on them a little like a bulldog, actually. He's a fascinating guy. So he was talking about a gynecological study that was handled in proteomics, and he made this comment. They ran all the controls on one day and all the cancers on the next day, Dr. Baggerly said. This is the worst kind of design when you are using a machine that can be subject to external factors, such as changes in calibration or mechanical breakdown. Pretty much all of the instruments we use meet that definition. So if you can avoid batch effects by running everything all together, that's powerful. If you are going to have to go to batches, try to use some of the strategies we talked about in the previous slide. All right. There are lots of different kinds of data analysis that come out of these once we've dealt with things like the data pre-processing, the normalization, etc. So I wanted to talk about some of these that come out of, some of these applications that come out of it. I'm going to focus on these top two. Class comparison. So if I have cases and controls, or if I have metastatic and non-metastatic cancers, or something like this, I have two classes that I want to compare and ask which genes are changing in expression between these cohorts. That is a really, really common use of RNA-Seq and microarray experiments. We also have class detection stories. Does everyone know breast cancer is not one disease? We call it breast cancer that as one great oversweeping statement. But in fact, breast cancer can present itself as a lot of very different kinds of conditions. Something like triple negative breast cancer is quite different than a case where a larger number of our therapies apply. So it might be that given a whole bunch of sick people who all seem to be sick from the same thing, may in fact be suffering from significantly different maladies. And so being able to discern classes of patients from experimental data is very valuable to us. I'm going to talk about a couple applications for that. If you're interested in this third case, asking which genes are useful in making a clinical decision, I would really suggest you take the clinical biomarkers module that'll be offered in August, because we're going to offer that for the third year. I think it's a good course, but we're not going to talk about it today. All right. So we're going to talk about clustering, because clustering is a really valuable technology that we use in all kinds of different data sets. In this case, we are going to do a data mining technique to infer a hierarchy that joins together a group of data points, like the little phylogeny we have up there to the left. It requires a distance metric. In other words, if I have data for sample A and data for sample B, I need to be able to compute in some way, are these close together or are these far apart? That's a distance metric. If you hear someone talking about Euclidean distances or Manhattan distances, they're talking about this distance metric. All right, now I might use an agglomerative approach. Everyone say agglomerative. Agglomerative. Isn't that delightful? Agglomerative. That's a very unusual word. This means we are grouping things together. So you might say, if I have a short distance between 2 points, I'm no longer going to treat them as two distinct points. I'm going to group them together, and now they have been agglomerated. All right. I might instead use a divisive strategy. I see that these 2 points are very far apart, therefore I'm going to slice between them and say they are two different groups. So one is so you can think of this as clumpers and splitters, right? This one is clumping data together that are more similar. The second one, divisive, is that we're trying to cut things apart that are far apart. We often create this thing called a dendrogram. That's a model of which we have at the upper left there. What's dendro? Dendro is fingers, right? So dendrogram, we're making a diagram of different fingers to show how these things can be split. Now, I wanted to note that the point I'm making at this last one looks very simple, but in fact it's quite messy. Most dendrograms can be cut at various levels to produce the desired number of groups. If you look at the figure at the top, you might say, well, it's quite obvious that A and B form one group, while C, D, E, and F form another group. That's one way to read it, right? Would it be appropriate to say that dendrogram shows that A, B, and C are one group, but D, E, and F are a separate group? Not really. Because if we're just drawing the line horizontally across there at some height, we can get to 2 groups, or 3 groups, or 4 groups, or 5 groups, depending on how low we set that line. The dendrogram defines basically where which group identity results from any of those cuts. All right. In practice, when we look at data from microarray or RNA-Seq experiments, we often see these checkerboard patterns. And I want to try to explain where those come from. We just had one slide about clustering, and I realize that this is a nasty thing to spring on you, but we can do clustering in multiple dimensions at the same time. In this case, we have different genes. Every one of the rows here represents a different gene. So we have a bunch of up values, and we have a bunch of down values for this particular gene. Across the horizontal axis, each of these different vertical stripes represents a different cell line, So in this cell line, described by the column, we have this gene, and we can observe that it was up or down relative to the main. So in this case, we have clustered together which genes show similar patterns of expression on the y-axis, and we've clustered together which cell lines show similar patterns of up or down in their genes on the x-axis. This is biclustering. There are lots of ways to go about doing biclustering, and some of them work particularly well and some work particularly badly, but you'll see quite a diversity of them. I don't remember if I left the slide in there about them. Oh, I did, good. All right, so we have things like Bayesian biclustering, or the Plaid model, or CPB, or cubic. All of these are different strategies people have put forward to do the biclustering. If you see something appearing in the journal Science or Nature, are you sure that the clustering approach they chose was absolutely right? Do not be fooled by the impact factor of a journal, right? Every team of people may have a preferred software approach that they use, but it might be that they would have seen completely different biology had they just switched from Plaid to cubic, or cubic to CPB, or BBC, or whatever. Different clustering approaches can produce very different patterns of association. So it's always worth keeping in mind that what you see as these presented models in RNA-Seq and microarray papers are kind of a conditional way to look at them. They're intended to be general guides to what was observed, but they are not the final arbiter of truth from that data set. I want you to just note that there's always multiple ways to do stuff, and which one someone chose may often be quite arbitrary. It may simply say that they've used that one a lot in the past and they have a pretty good sense of whether they're making errors with it or not. Oh, that sounded like a good one. Okay, well, we're going to come now to this differential expression thing. So I have a set of probes down the left side of this graph. So these, though each of these, say 1494 underscore F underscore AT, these represent a probe from a microarray. And now I have 2 cohorts. One in which we stimulated the cells with no HNE, and others in which we stimulated the cells with 60 micrograms or whatever of HNE. This is it doesn't matter. I mean, the case and cohort identities in this case don't really matter. So if I'm looking at a particular probe, I'm in this case maybe looking at a particular transcript, and I have these values that were observed for it. So a 6.35, a 6.23, a 6.20. On the right, I have 6.42, 6.50, 6.27. We want to ask the question, of all the genes we looked at, which ones are most differential between these 2 classes? So, how do we get there? Very, very frequently, we make use of the t-test because a lot of people know how to use that, and they assume that if they know how to use that, they're going to use that in replacement of anything else that they might use. T-test is not asking the question, are the cases and the controls of different populations? They're not doing that. The t-test is instead evaluating the hypothesis, the null hypothesis, that all of these values come from one distribution. When you get a p-value from t-test, it is asking how likely is it that the data would seem this different or more by random chance if they all came from one distribution. So we use it as a difference test, and we say, well, if the p-value that comes out is below 0.05, these and these are different, are proven different. Please don't say something like that, because it'll make me just twitch at the honors presentations at the end of the year. I frequently ask people, where did that p-value come from? Students don't like being asked that in the presentations. Doctoral students don't like being asked questions like that. So be aware that at the end of the year, hopefully I'm able to attend the honors presentations. If you have a p-value and don't know where it came from, you are in trouble. Just be aware of that now. All right, so the null hypothesis is the model that's being tested. Given that these data all come from the same distribution, what is the probability that the results would be this dissimilar or more? Okay, now, does everyone know the relationship between t-test and Guinness beer? You don't? I love getting to tell this story. But I'm a little sleepy, so I'm going to tell the short version. Is that okay? All right. Way back at the start of the 1900s, biostatistics was really gaining a headwind. They were doing an awful lot of stuff. Now, I taught biostatistics a couple years back here, and I taught it as in kind of a rude way, I must say, because the beginnings of biostatistics largely came out of the eugenics movement. Does everyone know what the eugenics movement is? The eugenics people thought that we could better humanity by convincing people who were poor not to have kids and making sure that rich people had lots of kids. Yeah, it's right. It's real. That's real. Okay, so gossip was not one of these nasty eugenics creeps, but there were plenty of them, trust me. You feel free to watch the videos if you want to know more, but I'll just give you one of my favorite quotes. I don't remember the exact wording, but a luminary of the biostatistical field said, if the poor of the world are going to spam it, to spam the world with their get, we might as well get some value out of these children by making them work in factories. Right. So, eugenics, bad, right? How do you think the Nazis justified stuff? Because great academics were saying nonsense like that, right? Okay, so, all right, I'm gonna tap down about the Nazis and the biostats people and the eugenics people.
Things are warming up in here.
All right. Gets me really head up. All right, so, Gossett was often thought of as kind of a moderating influence among the biostatisticians of the time who were all getting into these really amazing arguments with each other. Some people were just particular jerks in this particularly jerky field. So, Gossett was working in the beer industry. He was working for the Guinness Brewery. And although in our wine farms, people might say, oh, it was 2005, very good year, and so on. They want each year to be distinctive in its delightful palate and so on. In the beer industry, they wanted each batch to come out the same way. And Guinness thought they'll hire a statistician to make it happen. They hired Gossett. Gossett put together some ideas about how to evaluate whether things were from the same batch or not, like the t-test. And he said, I want to publish this. So he went to his boss and said, can I publish this? And he said, no. Do you know why? Because Guinness considered it a trade secret that they'd hired a statistician, and they didn't want their competitors to also hire statisticians. So Gossett published under the name Student. We call it Student's t-test because Gossett was prevented from using his own surname by his employer. Okay, I'm sorry, I'm getting way too political in this lecture. But now you have some background story to how the t-test came about. All right. So, to talk about the t-test in detail is really very much its own lecture, but I will just state that if you are using the t-test, you had better know whether you are doing a paired or unpaired test, and you had better know if you're using a one-sided or two-sided test. T-test can allow for you to do an unpaired, it can allow for you to do a paired. T-test can allow you to do a one-sided or it can do a two-sided, but you better know which one is right. So let's start with paired. I want you to imagine that all the professors in the biotechnology department have decided to go on an experimental diet, right? Right. So I've now signed on to this, as a result, we're going to do a before measurement and an after measurement. And the before measurement is going to start from, let's say, the start of March, right? This is a good time for it, because we've all failed in our New Year's resolutions to get slim. All right, so we're going to write down the measured weight of everyone in the department, or mass in kilograms, if you like. Okay, fine, we'll do that. So I've now written down the mass in kilograms of every professor in the department. We are now going on our experimental diet. Our experimental diet is to eat wheatgrass and bean sprouts every day. It's a terrible life. It's a terrible life. All right, that's fine. We'll do it. So now, after 3 months of this diet, we again write down the mass of every professor in the department. We are doing a paired t-test. A paired t-test. The reason why is that my weight and Dr. Kirby's weight and Dr. Klein's weight and Professor Pugh's weight should not be compared to mine, right? So my weight before can be compared to my weight after because they both relate to me. All right? So in this case, paired is the only way to go. So it might be that if you look at my weight before and my weight after, I have a plus, right? Maybe I have a little box of those little marshmallow chocolate eggs from Easter in my office, and sometimes I eat one, all right? Sometimes I eat three. So it could be that wheatgrass and so on is not going to save me from myself. It might be that I have a positive value, right? So we don't do these in aggregate. We look at individuals going up or down in that case. That is a paired t-test. One-sided versus two-sided. What do you expect to result from this intervention? Do you expect weights to be different? Or do you expect weights to be higher? Or do you expect weights to be lower?
Lower.
Lower. Okay, so our intervention is of the nature that we only need to check if people's weights are lowering, not rising. If they're rising, well, I mean, really everything is messed up. But the hypothesis we're working with says, if you give professors wheatgrass or whatever I've said at this point, you expect that their weights will be lower because they're not eating donuts or chocolate eggs. Okay, so our hypothesis in this case specifies we anticipate a fall because of this intervention. Great. That is a one-sided interpretation. Do you know that if you tell t-test, I don't know whether things were supposed to go up or down, you've created a two-sided test and your p-value will be penalized by twofold? Right. So if you have a sided test, like weights should go down if this intervention is causing a change, you have a one-sided test, you have a much better chance of getting a significant p-value if you tell the software, I'm doing a one-sided t-test. And yes, even in Excel, you can tell it these data are paired, these data are one-sided. So even if you never do this in R, that's still going to be valuable to you. All right. Now, let us assume that we have used a statistical test to develop p-values for individual genes, and we have computed log fold changes for genes. Log fold changes. So I might have seen a signal of 1,000,000 for this gene in this cohort, and in the other cohort I saw a value of 200,000. 200,000 versus 1,000,000 is what fold change? It's five-fold down, right? 1,000,000 versus 200,000 is five-fold different. So that's a five-fold down change. So I'm going to take the log of that. So you see that we're going to use log base 2 fold change on our x-axis. Each of these dots represents a different gene. A gene that has a big change up appears to the right. Something that has a big change down falls to the left. Everyone good with that? You remember on the MA plot, everything was revolving around this zero line in the middle? This is the zero line here. So these are big changes up, big changes down. Now, we also have the negative log of p-value on the y-axis. So things up at the top had significant p-values. Things at the bottom did not have significant p-values. So how is it possible that something could have a big fold change but not have a significant p-value? We just talked about an example where I had 1 million counts over here and 200,000 counts on the other side. Surely that's differential, right? Not necessarily. Not necessarily. Because it might be that while it's typical to say that their means were 5-fold different, it might be that there was so much variance within this cohort and that cohort that I wasn't sure that was a significant difference. A big fold change is not the same thing as significance. Okay, so I want you to think, I wish I had a stick for, oh, there's a stick right here. Great, okay. No one tell them I use this for this. All right, so we already said that things to the left are down, things to the right are up. But I want you to think instead about something biologists often do. Biologists frequently say, even if it has a significant p-value, I don't care unless it's at least a 2-fold change up or a 2-fold change down. Why would they do that?
Lazy?
Because they're lazy? No, well, we're all lazy, but I would say that generally speaking, many biologists believe that a transcriptional difference that's less than 2-fold in one direction or the other is worth ignoring. Really. So this is a log base 2 fold change. Therefore, we're going to put in a vertical right here. See that? Anything left of this line is not a big enough change to be considered in this direction. And now I'm going to throw another line here, and I'm going to say anything to the left of this line is a big enough down change to be considered. So I care about the flanks, the stuff that's outside this +1 to -1 boundary on a log base 2 fold change. This is a biological rule. It is not, emphatically not, a statistical rule. But many biologists say if it's less than 2-fold change, I couldn't be bothered. Now, we're going to throw one more line across here. Right there. See it? All right. Anything below this line has an insignificant p-value. Even if it was a big fold change, it was not a reliable enough difference for us to call significant. So things that are above this line and outside, in those outside flanking regions, those are the hot spots. This is the stuff that's down that we're going to go chase after. This is the stuff that's up that we're going to chase after. Everyone sees that? What about these red dots? See those guys? That guy, that guy, that guy. They are significant by p-value consideration, but they fall within this plus or minus 2-fold change. Therefore, the biologists say this is not a big enough change for me to consider significant to chase. Okay, that's a volcano plot. That's how to read it. Now, multiple testing correction. This is a piece I care tremendously about, and yet I've saved it to the very last part of this lecture. I apologize for that. Each probe, in this case, each transcript, is going to have a p-value computed for it. Something that I really wish we were able to include in the R lessons is the final message. You can read, you can view that on YouTube from one of the other classes. But the point that I want to make here is that every transcript for which we're computing a p-value is a different test. It's a different test. The t-test and every other statistical test out there should produce, when there's no difference at all, a p-value that is below 0.05 1 in 20 times. What this means is that if you do 20 tests, 20 t-tests, of data that come from the same distribution with no difference, 1 in 20 times, the t-test will give you a p-value less than 0.05, even though there's no difference. This is a false positive, is one way to think of it. That t-test can give you a value below 0.05 for p, even if there's no difference in the distributions from which these data came. Okay, so I've already gone through that slide, sorry. So if you perform 1,000 t-tests, let us say that you're doing an RNA-Seq experiment and you only include the top 1,000 transcripts in it. If you perform 1,000 t-tests, 50 of them will look significant, even if there's no difference in the distributions from which those data came. That could be 50 false calls, right? That's worrisome. So how do we deal with it? We have two major approaches that we frequently use in multiple testing. So the first one of these, the Bonferroni correction, is from a very early mathematician, but it was first enunciated by Olivia Dunn, who was a female biostatistician who put together 2 and 2 and put together this really cool paper on this subject. So if you have done 1,000 t-tests, she says substitute 1,000 for the number of trials. Instead of looking for a p-value below 0.05, you should now look for a p-value below 0.05 divided by 1,000. Wow, that's a rough requirement, isn't it? But I want to point out that Bonferroni correction is intended to protect you against making any false claims. It's intended to prevent you from making any false positives at all. If you're able to tolerate a few false positives, then we have some much better news for you. In a case like that, Benjamini-Hochberg has a good correction for this thresholding. Benjamini-Hochberg seems a little odd. We're going to start by sorting all of the genes that we tested by the p-value that came out. So the very lowest p-value is at the top, the second lowest p-value is the second row, the third highest p-value is on the third row, etc. We then say, for the very lowest p-value we got, we're going to compare it to 1 over 1,000 times 0.05. If it is below that line, that's brilliant. It succeeds as something that we can call a positive. The next one gets 2 times as liberal a threshold. We double the value of the threshold that was applied to the first one. For the third one, we try 3 times the first one. So what you can see is that here we've got these 1s and 0s. These represent the different tests that we've run, the different genes. The lowest one had a p-value very, very close to 0. And you can see then that we have this slowly rising red line that represents a gradually increasing threshold in response to the Benjamini-Hochberg progression. This is quite different because in the first case, Bonferroni tries to protect you from making any false claims, for rejecting the null hypothesis falsely. In the second case, Benjamini-Hochberg is trying to limit the rate at which you accumulate false positives. So if you're willing to tolerate that 1 in 20 of the genes you claimed were differential is falsely claimed, Benjamini-Hochberg is perfectly fine for you. So these are rather different strategies that can be used for adjusting to the fact that we're doing lots and lots of statistical tests. Oh, you'll often see people call that FDR instead of the Benjamini-Hochberg technique. So if you see false discovery rate, it's very often implemented by the Benjamini-Hochberg approach. It's not the only way, but it's one way. All right, now, things come back from this that we frequently hear when people are complaining about multiple testing correction. Some people will say that after they've used multiple testing correction, all of their hits vanished. Therefore, they can't use multiple testing correction. So I would note that it's possible that the null distribution was actually right. Sometimes your PI sends you on a project sure that these genes are going to be different from each other when you do the test, but the PI is sometimes wrong. Sometimes we do experiments where we expect to find a difference, and we don't. What if my wheatgrass that we force feed all the professors here, wheatgrass, what if that doesn't actually reduce our weights? My hypothesis might be wrong. So just because I didn't get a significant p-value and expected I would get one, doesn't mean that I did the experiment wrong or that I did the data analysis wrong. It could be that the whole hypothesis upon which this was based in the first place, the biological premise, was wrong. The next is that sometimes the experiment was underpowered. Have you noticed an awful lot of the research we talk about has 3 replicates of this class and 3 replicates of that class? Do you know why that happens? Is 3 a magical number in statistics? Not particularly. In general, people choose to do 3 replicates because they think that's the least you can do a t-test on, and they think to themselves, that that's all I can afford, or that's all the time that my grad student has to perform these experiments. Frequently, this means we have an experimental design that is underpowered. That even if the difference really existed, the experiment, if completed, has very little chance of detecting this difference. So it may be that the experimental design in the first place was not worth running. I have to talk a little bit about reality here. And that is that when you seek to publish your research, you will be up against some stresses that people at Harvard University will not be, or that Caltech will not be up against, or that MIT won't be up against. The reality is that people from famous laboratories who produce their research frequently make mistakes in their data analysis, and the reviewers let the mistakes slide, thinking, well, they're in a famous laboratory. A famous laboratory obviously wouldn't have made this mistake. But if they receive a paper from a university in South Africa that they've never heard of, this is the reality. They're going to be watching the work with hawk eyes. Famous laboratories are able to get away with mistakes in their data analysis that lesser-known laboratories are not. So just because you see that famous lab, Professor Dr. such and such, has published without using multiple testing correction, does not mean you could get away with the same mistake. That professor has made a mistake. Their results are invalid, but they still got published for it. And you may not be able to count on the same happening for you. So this is awful, and all I can say is that don't be one of these morons. People who have very rich and wealthy labs make screamingly bad mistakes all the time. We are all of us, including people at rich and wealthy labs, are subject to the same critique of science. And there will be muttering campaigns about certain papers that have been published in my fields for decades on end. You can't take advantage of being in a famous laboratory, perhaps. But we can only do the best science we can. That means using multiple testing correction, because that's the right thing to do. Sometimes people don't even realize that multiple testing correction is required. What if you did 5 groups, maybe apples, pears, bananas, limes, whatever, and you then said, well, I know how to use t-tests, so I'm going to compare pears to apples, and pears to oranges, and pears to limes, and pears to bananas. And then I'm going to do limes to bananas, limes to etc. I mean, all of these different connections. With 5 different groups, we have 10 comparisons, which means that even if all of these fruits have exactly the same properties, you have a 50-50 shot of publishing a false positive just because you did so many comparisons if you don't use multiple testing correction. For a situation like this, using ANOVA would have been appropriate, because ANOVA is designed for multi-part, multi-level comparisons. I think Ronald Kosa has a really valuable rule, and if you all just memorize this, I'm going to feel happy. If you torture the data long enough, it will confess. You might think that the way to please your advisor in the course of your honors project is to keep trying different statistical tests until one of them gives you a p-value below 0.05. Please don't learn that lesson. That is a terrible, corrosive lesson. If your study turns out to have a negative result by a previously designed statistical approach that you feel sure is the right way to go, the answer is not to go shopping for the metric that will give you a significant p-value. Really. Really. That's a hard lesson. We all want to publish in Science and Nature. Of course we do. We don't get there by fudging the numbers. Remember Kosa. If you torture the data long enough, it will confess. Lots of lessons here. Microarrays have given way to RNA-Seq for gene expression measurement because you can do a lot of things with RNA-Seq that you can't do with microarray data. But in all of these cases, statistical considerations are going to run the show. Bioinformatics has great workflows for all kinds of things that we've talked about today. But in all of these cases, statistical considerations are going to be a very big deal. When you see a bicluster in a paper, when you see a volcano plot in a paper, I hope you understand how to interpret it. We walked through those in some detail today. We also talked about MA plots. Some people are studious about presenting those too. Systems biology, not just microarrays, not just RNA-Seq, frequently produce situations where multiple testing is a problem. Bonferroni and Benjamini-Hochberg false discovery rates are very valuable techniques for handling this. And that is our quick introduction to gene expression. Thanks, everybody.